The overall goal of this procedure is to obtain image series from immobilised human sperm labelled with a fluorescent calcium reporter dye. First, motile cells are isolated from seminal plasma by swimming them up into incubation medium. After capacitation, cells are labelled with fluorescent calcium indicator, then transferred to a perfusable imaging chamber which is mounted on an inverted fluorescence microscope. Calcium ion concentration responses are captured as a time-lapse image series and analysed to extract single-cell responses.
Hi, I'm Aduen Morales-Garcia from the laboratory of Dr. Stephen J. Publicover in the School of Biosciences at the University of Birmingham, UK.
I'm Katherine Nash, also from the Publicover lab.
And I'm Jennifer Morris from the Publicover lab.
Today we'll show you a procedure for imaging intracellular calcium responses in individual human spermatozoa.
We use this procedure within our laboratory to study regulation of sperm activities using stimuli derived from the egg and the female tract.
So let's get started.
Store semen samples at 37 degrees Celsius for no more than 30 minutes from the moment of collection. To isolate motile cells from the seminal plasma, pipette 1 ml of Earls balanced salt solution supplemented with 0.3% BSA, abbreviated as SEBSS, into a series of 5 ml polystyrene round-bottom tubes. Gently underlayer the SEBSS in each tube with 0.3 ml of semen.